All right, here we begin Chapter 7, Section 3. And here you have just two objectives. Should be Section 3 there. But don't let that fool you. This is probably going to be the hardest part of the chapter. And so for the audio portion, there's probably going to be two parts. All right, so what we're going to be talking about here is the main catabolic pathways that occur in all cells, whether we are talking about eukaryotic cells or prokaryotic cells. And the point of these metabolic pathways, in terms of the catabolic pathways that you see here, is the production of energy. All right, cells need energy. They can't do anything without energy, and cells have to be able to produce energy. And so this figure is an overview, right, big picture, and it shows you some different scenarios that you might find within cells. So on the left, you have aerobic respiration. In the middle, you have anaerobic respiration without oxygen, and then on the right side, you have fermentation. All right, so all three of these are going to have one common feature, and that's going to be the process of glycolysis. Glycolysis doesn't produce a whole lot of energy, all right, just 2 molecules of ATP. And it's going to produce 2 molecules of ATP regardless of what overall pathway you're looking at. And then aerobic and anaerobic respiration feed into the Krebs cycle, which feed into the electron transport chain. And what the difference is here is whether or not oxygen is available. And if oxygen is available, then it's aerobic respiration. And at the very bottom, right, you have the electron transport system, and it's going to use oxygen as the electron acceptor. And in that process, make water. In anaerobic respiration, water is not available, and so then you have some other component, all right, these options down here, that are going to be the oxygen or the electron acceptor and produce other compounds besides water. All right. Aerobic respiration in general is going to produce the most amount of oxygen. Anaerobic, depending on the exact nature of the cell and what is available, it will give you a range of 2 to 36 ATPs. And then fermentation doesn't give you a whole lot of energy. However, it gives a cell other metabolic byproducts, and some of these can be used for consumer benefits. All right, but still not a whole lot of energy, but cells will still use it. All right. So when we talk about aerobic respiration, that's really what we're going to focus on, is cells when they have oxygen available. All right, and glucose is going to be one of those very normal nutrients that cells have a lot of, or that glucose is available to those cells. And glucose is just the go-to molecule when you talk about aerobic respiration, because it occurs a lot, it occurs very quickly, very efficiently, and it's just easy. All right, so aerobic respiration, you think about respiration and you think about breathing. Humans respire, all right? We pull oxygen in for our cells, and then we blow off CO2. All right, when we're talking about cells, particularly bacteria here, aerobic respiration is a series of reactions that converts glucose to carbon dioxide, and then allows the cell to recover significant amounts of energy. All right, that significant amount of energy is in the presence of oxygen and utilizing only 1 molecule of glucose. And you need to remember that a cell probably, if it has glucose available to it, it's probably not just 1 molecule of glucose. It's probably flooded with glucose. And so then the cell can pull in many molecules of glucose and go through the process of aerobic respiration for however many times the number of glucose molecules are available, and then for each molecule of glucose can produce 36 to 38 molecules of ATP. All right, this process has different steps, right? Glycolysis, the Krebs cycle, and the electron transport chain. Anaerobic respiration, right, as I mentioned, it still involves the same series of reactions. However, oxygen is not available, and so it's going to use these different molecules for oxygen to bind to, all right, in terms of those final, or for those electrons to bind, and it doesn't produce nearly as much energy. Fermentation is an incomplete oxidation or breakdown of glucose. Oxygen is not required, and in many cases is not available, and so then organic compounds are the final electron acceptors. So here is a very simplified version of glycolysis. And when I say very simplified, if you were taking a biochemistry class, all right, an upper division, senior level college biochemistry class, there would be an entire chapter on just glycolysis, right? Not just part of one section of one chapter, which we're dealing with here. It is very complex in terms of the different steps, the number of enzymes that are necessary to go through the different steps of glycolysis. But what we're going to do is really focus on the big picture. And for all of these different steps, what I want you to be mindful of are the input and the outputs of each reaction. So with glycolysis, you're going to start with a glucose molecule. And for simplified picture diagram effect, glucose is a 6-carbon molecule. All right, then there are going to be other reactions that occur, and in this specific step, 2 ATP molecules are used. All right, so to get the process of glycolysis going, you have to give it just a little bit of energy, and then it does its thing. And here you have up top here, you have a linear reaction. All right, and then down on the bottom, you see where the linear reaction diverges, and you have 2 reactions going side by side. The end product of glycolysis is the production of 2 pyruvate molecules. All right, and then also at this point, the reaction will yield 4 ATP molecules. So then, the total energy yield with 1 molecule of glucose are 2 ATPs and then 2 NADH molecules. The NADH molecules are important because those are what's carrying the electrons, right? In the form of the hydrogen right here, it's going to carry those hydrogen over to the electron transport chain. So end product, 2 pyruvate molecules, and then 2 ATPs and 2 NADH. All right, the 2 NADH are not important in terms of specifics for glycolysis, but rather the understanding that these NADH molecules are going to be used further down the line. Now here's the next figure of the Krebs cycle. And what I want to point out, and this is kind of old school material, doesn't always get included in the discussion about aerobic respiration. All right, so here's the end product of glycolysis, where you have the 2 pyruvate molecules. The pyruvate molecules are going to feed into the Krebs cycle, and because you have 2 pyruvate molecules, then you're going to complete 2 cycles of the Krebs cycle for every 1 molecule of glycolysis or glucose. However, there is this step that is referred to, again old school, as the transition step. And that's because pyruvate doesn't directly feed into the Krebs cycle. It has to be converted into acetyl-CoA. And so this transition step results in 2 acetyl-CoAs, one for each pyruvate molecule, and then another 2 NADH molecules. The NADH moves over into the electron transport chain. All right, so you have the transition step, and you have produced the 2 acetyl-CoA. So input for the transition step is pyruvate, output is acetyl-CoA. So then the Krebs cycle here, it's a circular reaction, a circular series of reactions where acetyl-CoA comes in. It is acted upon by different enzymes. CO2 is released. GTP is released. And again, GPT can actually be used as a form of energy similar to ATP, but it's not at high concentrations, and it's not quite as readily used as ATP is. Right, and as it continues to go through this cycle of reactions, you have more CO2 that is released, and one of the final steps is the production of the oxaloacetate, which then feeds back into the reaction when more acetyl-CoA comes into the mix of things. And so yield, really, the purpose of this acetyl-CoA is the generation of NADH3 molecules and FADH2. Right, the byproducts are the CO2, yes, then cells are going to get rid of that, but really the important thing is these carrier molecules, the molecules that are going to carry the electrons over to the electron transport chain. So then the electron transport chain is a series of proteins. All right, it's a chain of special redox carriers, so those reduction and oxidation reactions that receive the NADH and the FADH2 molecules that have been generated by glycolysis, the transition step, and the Krebs cycle. And so what happens is this series of reactions takes place from one protein to the next, and the electrons get passed sequentially and orderly from one protein to the next. And this is a highly energetic reaction, and it transports hydrogen ions from the interior of the cell to the exterior, and then they come back through a ATP synthase, and the oxygen accepts the electrons and the hydrogen, and it forms water. And in the process of that creates ATP. Alright, so this is the diagram of what's going on. Alright, you see these series of proteins here, kind of a rainbow color of proteins, and they are in a sequential pattern. They have specific names. That is just a lot of detail that we're not going to work through. But they are found in the cell membrane. Alright, so we're talking about prokaryotic cells here. ATP generation occurs at the cell membrane, whereas in eukaryotic cells, ATP production is found in the mitochondria. But prokaryotic cells, by definition, do not have mitochondria. So, NADH and FADH2 come in, bring that hydrogen. The hydrogen gets pumped across the plasma membrane. The electrons get carried from one protein to the next. The hydrogen come back through the plasma membrane, through this ATP synthase. And ADP, along with phosphates, which they don't show up here, allow the formation of ATP. All right, simply linking ADP to that third phosphate group, and now you have ATP. And then you have oxygen here for aerobic respiration. The hydrogen comes in and bonds to oxygen, and now you have H2O. And then in anaerobic respiration, all right, you have these other molecules. So that is where we're going to end part 1 of section 7.3.